Okay, welcome to the next session. And I'll warn you, the Italians are coming. We have two speakers. Our first speaker, and I have tried very hard to get them into a fight. So we're gonna work on this in the questions. So see if you can think of something that'll put them on the opposite sides of any fence. We have, first of all, Stefano Manegatti, who was born and raised in the northeast part of Italy. And of course, Michael's family was from Naples, the opposite side of Italy. And to make things worse, he was raised in an Italian family in New Jersey. So they should have plenty to argue about, at any rate. So keep your eyes open. Stefano did his bachelor's, his master's, and his PhD here in chemical engineering, chemical and biological engineering here at NC State. He worked with Ruben Carbonell as a PhD student, and NC State was so impressed with him, they did something that you don't hear about very much. They offered him an assistant professorship before he even left to go do his postdoc. They guaranteed him a space when he came back.
You're probably afraid, I don't know, retaliation.
Saul and veto. Anyway, so he went to work with Samir Mitragotri at University of California at Santa Barbara. Samir last year was one of the youngest people ever inducted into the National Academy of Engineering. So they did some fantastic work together. And you're going to hear some very interesting things about what Stefano is doing now with peptides and peptide mimetics. Stefano?
Thank you so much, Frances. We can try to find as much as we, in the extent that we can, but I really couldn't think of a more congenial collaborator than Mike. We'll try to find something to fight upon. So today, as Frances said, the topic of my presentation is going to be peptides and peptides mimetic, really the new front of peptide chemistry and peptide biochemistry. So what you see here is me or us playing in the lab with peptides. Now it's really a lot of fun. It really resembles something wrong. See my Alzheimer. Playing with peptides is really, oh no, I can tell. Playing with peptides is really a lot of fun because it truly resembles playing with Lego. I mean, you probably have played yourself, you have seen your kids playing with Lego. They really look alike because they're modular, they can be combined in a lot of ways, and there is really no limit to the geometries, the shapes, the size of the things you can construct with them and how fascinating they can be. Even the single blocks look alike. They have a positive side and a negative side that they fit into each other, and they basically can create, as you see here in the picture, they basically can create a chain of colored objects. So instead of having a piece of Lego, a little single brick, now we play with amino acids. Amino acids have two handles that basically connect with each other, and they have something that defines their unique features, their chemistry, their property, this side chain, what is technically called the side chain functional group, which defines, if we want, the biochemical color and shape of the single building block. So truly playing with peptide chemistry and peptide mimetics is truly like playing with Lego, and there is no limit to the array of geometries and shapes and size that you can really achieve. And what I want to do today, I want to take you through a sequence, a sort of a quick perusement of our menu, so you can actually see, we can actually see together the wonderful and so diverse things that can be done with peptides. It can have impact on industrial bioseparation, it can have impact on biomedical engineering, it can have impact on polymer science and chemistries, and so on and so forth. So the first topic for today the first part is on alpha peptides and their applications in bioseparations and biomedical engineering. Let's start with separations. By separation, by bioseparation, we mean purification of therapeutic molecules that are biologics. So the purification of protein therapeutics. One protein therapeutic of interest is IgG, immunoglobulin G, which is utilized, monoclonal antibodies are utilized for fighting a variety of diseases, but most frequently cancer. Our peptide, the peptide that we have identified, and here I have, the example of 3 linear alpha peptide sequences. They bind IgG, they bind the immunoglobulin G in the lower part of the Y-shaped molecule, the region that's called PFC, with a certain good affinity, with a decent affinity. If we quantify the dissociation as a measure of the strength by which the peptide ligand binds the IgG biomolecule, this dissociation constant is very, very low, which means that these 2 molecules tend to like each other and stay with each other a lot. So I asked you to memorize this number here, 10 to the minus 5 molar, because it will make sense while we progress through the presentation. But anyway, 3 linear alpha peptides that like IgG a lot and can be used for its purification. How do we do that? Well, we couple, we attach, we conjugate those linear peptides on the surface of chromatographic beads. We pack these beads into a column, into a vessel, and we fill this vessel with a mixture of our target protein and protein impurities. So a mixture such as that could be human plasma. It contains IgG along with other molecules. It can be a recombinant cell culture fluid that contains IgG together with a completely different variety of molecules, and so on and so forth. We can purify IgG from milk, we can purify IgG from plant extracts, really from a variety of sources, all using this one simple technology, this one simple affinity-based technology. What's interesting about these peptides that we identify, these peptide ligands, is that they bind all the classes of human IgG, unlike, for those of you who are familiar with the bioseparation, unlike with protein A and protein G, which are the gold standard in the industry for bioseparation, our peptide is really able to bind all the 4 subclasses of human IgG, but it doesn't stop there. It also binds animal IgG. And why is the purification, the separation of animal IgG from animal serum important? Well, because these molecules, these animal IgGs, are today utilized to build diagnostic assays. So it is nice to have an affinity technology that allows us to purify at the very large scale biomolecules that are needed to construct bioassays, but cannot be currently purified or are difficult to currently purified with the separation technology that's out there. All of this enabled by a simple linear little string of 6 amino acids, an hexapeptide. Now, to give you a visual confirmation of this, imagine human serum, which comes with its own complexity of protein, and the target molecule, which has a light chain and a heavy chain here. And you see that after passing through the process, after passing through our affinity chromatography process, the peptide-based affinity separation process, the IgG becomes almost magically, in a single pass, hence the magic, becomes almost magically purified in a single pass. And what holds for human serum holds for other kind of serum. You see here serum obtained from camels. Camels' antibodies are very interesting. They're very hot today, a very hot biomolecule, because it's a single chain, hence it can be expressed, produced way more easily than fully human monoclonal antibodies in the industry. It is easy to express, but it's not easy to purify because the current technology doesn't have the handles to pull camel IgG out of the complex mixtures. This ligand actually has the ability to get camel and llama single chain antibodies out of a mixture in a very pure manner. Well, one say, are linear peptide always good? No, they're not always good. They're good when the concentration of your target molecule is high enough so that those little linear handles can actually grab it out of the complexity of a protein mixture and return it to you during the elution process. But linear peptides are not always enough. Sometimes we really want to have a higher affinity, a stronger grabbing handle. And to that purpose, we go from linear peptides, so moderate affinity, to cyclic peptides that have a much higher affinity for biomolecules. There's a whole thermodynamics explanation of that, which we can discuss after the presentation. But trust me and let's keep going. The cyclic peptide actually do a better job at capturing low concentration, owing to their stronger affinity. On the other hand, finding linear cyclic peptide is not easy. It's not easy to find them. It's not easy to sequence them. So we have devised a method in which we prepare libraries, we prepare ensemble of cyclic peptides, we screen them against our target biomolecule, and after we have identified, we actually split them open, we break them open, we linearize them, and from that point on, the technology is easy for sequencing them. So we have devised this chemistry and mass spectrometry-based system for easy identification of cyclic peptides. So that gave us the possibility of really starting looking into ligands to separate really low abundance molecules from complex fluids. And one of them that we particularly liked is erythropoietin. Erythropoietin is the protein that regulates the production of red blood cells in our body. And it's found either in our blood or in recombinant fluids at a very low concentration. Pulling it out requires high affinity, hence cyclic peptides. We did a selection, we did a screening process, we came up with good candidate sequences. You see this one that I've boxed for you. We have done a whole large and comprehensive work of characterization, seeing how tightly and how much protein these ligands could recover. And the results are actually interesting, very interesting. The complexity of the solution in which erythropoietin is found is fairly high. It contains, of course, all the protein impurities or competitors that are found in our blood. But the ligand is actually able not only to return it to us at a much higher concentration as compared to the feed sample, but also to return it in at an almost complete or quasi-complete purity, which is a great way to extract really high-value molecules from our blood. The reason why it can do such a fantastic job is because its affinity is actually much higher, at least 2 orders of magnitude higher. You've seen before the 10 to the minus 5 number that I asked you to retain. Now we have a 10 to the minus 7 number. 100 times stronger affinity simply because we went from linear structure to a cyclic structure. And again, there is solid thermodynamics behind this. Now, what does that mean? Well, that basically means that if you do a refined search of your molecule, of your peptides, of your cyclic peptides, and you do a considerable work of engineering of your ligand, of improving your affinity, you can actually have a much better handle to recover molecules that are much lower concentration in a complex fluid. So, we look at where in the target molecules our ligand bind. And we start doing mutational analysis, and we start doing lots of tricks, computational tricks. When we synthesize, the ligands that we predict would work better, and we test them, we actually find that they work really well, and they can separate, erythropoietin at a very low concentration. So what's the standard? How do we move the standard? Well, we move the standard from, the little linear peptide that was binding monoclonal antibodies at a concentration of 1 to 10 milligram per milliliter or gram per liter, to a cyclic peptide that can bind molecules at 1,000 times lower concentration. And this is all done with a combination of theory and experiments in our lab. And just to show you a taste of what we do, we really combine experimental characterization and computational characterization to really refine our sequences. One more thing that I want to show you today is, well, if they can capture protein in solution, why can't they target protein in solution? Of course they can target protein in solution. And using the same technology, we have developed things that are good not only in the bioseparation world, but in the biomedical world. For example, we have produced synthetical nanoplatelets, which means soft nanoshells produced through a layer-by-layer, fairly simple technology, decorated with linear peptides. These shells, decorated with linear peptides, can do three things that platelets do very well. They can grab the von Willebrand factor and the collagen, which gets exposed when your blood vessel is severed. So they can do targeting, they can do blocking. And they can also do recruiting of other platelets, of real cell platelets from the bloodstream. So, one fully synthetic, peptide-decorated little nanoparticles doing the same job as a normal platelets would do. And to show you a demonstration of this, we have done a microfluidic characterization of our synthetic platelets and an in vivo characterization of our platelets. Now, just focusing on the important stuff, we see that in our microfluidic characterization, synthetic platelets and natural platelets tend to overlap at the wound site, at the synthetic wound site. Then, therefore, as you see here, the shape of the green and red color are the same. The red color are the natural platelets, the green color are the synthetic platelets. Hence demonstrating that our synthetic platelets co-localize together with the natural platelets at the wound site. They do as good as a job. And if you take a little mouse, and you pre-inject the mouse with our synthetic platelets, and allow me to say this, cut his tail, the mouse will bleed between 10 and 20 times less, or the bleeding will stop 10 to 20 times before, as compared to a mouse that has not been supplemented with synthetic platelets. So once again, the powers of peptides, decorated, attached on solid surfaces, targeting proteins at the very low abundance in a complex solution. One more thing, permeation through skin. Skin is a very tough barrier. It's almost impossible to overcome it unless you're trying to permeate very, very small molecules. So we have embarked ourselves in a search of, once again, peptides that would enhance the permeation of molecules that otherwise would not be possible to deliver through the skin and actually take them effectively across the skin barrier. We do this because eventually we want to create a skin immunization by topical spraying. But what I'm showing today is the preliminary work that we have done with new cyclic peptides utilized for delivering biomolecules through the skin. And we have seen, for our model drug cyclosporin A, which is an immunosuppressant and an antibiotic, we have seen that for cyclosporin A, we have been able to deliver almost 20 times more than the drug alone, just by a little supplementation of, at very low concentration, of our skin-permeating peptides. And one could say, well, granted, they take your molecule and they take them across the skin. They do what the drug alone could not do, but are they toxic at doing it? Can they elicit irritation of your skin? Well, actually, we tested that too, and we have seen that in a very wide range of skin-permeating peptide concentration applied on the top of skins, we see no irritation and no toxicity. There is no immunogenic response, there is no irritation as compared to classical irritants such as SDS, and so on and so forth. So not only they're effective, they're also safe. Well, now one can say, fine, I understand all of that, but I'm a biochemist and I understand biochemistry, and I understand that when you have a peptide into a biological fluid, that biological fluid is also going to contain stuff that's going to chew your peptide. Proteases, peptidases. How are these peptides going to survive in that tough environment? Skin has proteases, the blood has proteases, there are proteases in the cell culture suprases, there are proteases in every biological fluid. How are your peptides going to make it in that tough, challenging environment? True question, good question, here's our answer. The answer is, well, it's true. You can have sites where enzymes can cleave your peptides, but we can actually mock those enzymes and modify the sequence of the peptide using non-natural amino acids so that those peptides become not recognizable by the agents that would otherwise chew them up. And indeed, we have observed that You know, you remember our own little linear IgG binding peptide? Well, we have observed that that peptide, modified in the opportune way, modified in the correct way, not only retains its affinity for the target, but it also gains resistance to enzymatic attack. So these peptides become like Robocop. They do what they want, regardless of who they are surrounded by. And to give you an example, so we have done, you know, some simulations, of course, because we wanted to see how the binding pocket on our target protein would change by modifying the sequences of our peptide ligands. We have seen that, you know, the modification of the site of interaction is not dramatic, so they tend to bind at the same site. They tend to bind with a fairly similar affinity. You know, you can see the slope of these two curves are practically the same. They tend to bind with the same affinity, but they resist cycles, many cycles of proteolytic attack. One more thing that they do, which was some sort of an added value and a good surprise for us, is that they actually increase the selectivity. If you see the purity of the antibody purified from plasma using the old peptide, the natural peptide, well, yeah, the affinity, the selectivity was pretty good, but there were some small impurities here and there. Using this modified amino acid, we're actually able to further tune its selectivity for our protein target, for our antibody, and the peptide ended up not only being more biochemically resistant, but it also ended up being more selective for our target protein. And in fact, you see here many less impurities as compared to the parental sequence. Well, one could say, it's all interesting, you know, non-natural amino acids are giving you a further handle to increase the biochemical resistance and increase the reach of your ligands. Reach in the sense of affinity and selectivity. But is that it? Well, no, it's not even it. Because we can actually think of completely different or almost completely different variants of peptides in terms of more inherent modification of their backbones. Years ago, a very smart guy from California came up with a very simple synthetic method to produce what is nowadays universally known as peptoids. How do they differ from peptides? Well, they differ because the side chain groups, you know, those colored things that make our amino acid look like the Lego, the side chain groups, which define the biochemical color of the amino acids, instead of being attached in this position, are actually attached in a different position. Well, one could say, well, big deal, looks the same to me. Well, that small modification gives higher selectivity, and I can tell you why. Higher biochemical stability, and I can tell you why. High tissue permeability, and I can tell you why. And no chirality. So let's look into them. High selectivity, why is that? Well, because of the way the side chain functional groups are appended, these backbones, unlike this, can actually entertain less non-specific interactions with other protein in solution. Why is that? Because they look like protein, but they ain't protein. And so they entertain many less of those protein characteristic non-specific interactions that protein can entertain or peptide to protein can entertain with each other. And so they have a higher selectivity. Higher stability because they look like peptides, but they're not peptides. And so the peptidases don't know what to do with them. They don't know how to chew them up. Higher permeability because by being a tiny little greasier as compared to peptides, they can actually permeate tissues, skin, and organs with a higher efficiency. So they make better drugs. They tend to make better drugs. They have been demonstrated to make better drugs. No chirality means that, here we go a little bit more into the specificity of the chemistry. No chirality means that there are no, let's say, batch to batch or synthesis to synthesis diversity, let's keep it at this level, between peptoids instead of peptides that do show chirality, and therefore they show imperfections and batch-to-batch variation, between even within the same sequence. What do we do with peptoids? How do we discover peptoids that target a certain class of molecules? How do we discover peptoids that target a certain class of interesting biomolecules targets? Well, We do a combination of things. We do a lot of experiment, especially we do NMR and CD, circular dichroism, to understand, to gather some preliminary data about their structure. And then we feed this data and we feed our models into a computer that runs atomistic simulation and then coarse grain simulation and really tries to understand how these molecules, this new class of molecules, actually arrange in the space, how they acquire their secondary structure, and therefore how they can entertain specific relationship with larger biomolecules. So by running these simulations, you know, with supercomputers, we actually are gathering a better idea, because again, they are new molecules. Very little is known on how they organize in the space, but we're getting a better and better idea on how they aggregate, how they display themselves in a liquid, how they interact with proteins, and after we have done these simulations, we actually synthesize model molecules and we recharacterize them in a sequence of loops, so that we can achieve our dream. And what is our dream? What is our main goal with peptoids? If you look in the pharmacopoeia, if you look in the literature to what the bioactive peptides are, the peptides that have drag value, that have ligand value, you will see hundreds of molecules that are already discovered. I bet you anything, if you come up with a biological target and we spend 15 minutes Googling it, we will find a peptide that binds that molecule, whether it's for used as a drug or used as a ligand to purify that molecule from a complex source. So why in the world would we have to rediscover peptoids for these ligands? Peptides that would make better drugs, with better permeability, with higher residence time in our body, and so on and so forth. We don't have to rediscover them, because we are actually working on an algorithm that would translate known bioactive peptides into bioactive peptoids that do the same but better. Again, with higher proteolytic stability, with higher tissue permeability, with higher half-life in the body, and so on and so forth. And to give you an example of this, let's go back to our hexapeptide that binds IgG, our short friend that binds IgG. Well, we have translated it into its own peptoid variant. It looks similar, but not quite there, because the software had to figure out how to make it similar, how to behave similarly, without being the same thing, without actually having exactly the same three-dimensional structure. So you see, some of the chemistry changes, some of the side group changes. These are things that the software now is figuring out for us. The handle, the chemical handle that goes from one separation to two units of separation, the hydrogen bonding which is reformulated, and so on and so forth. But guess what? Simulations show that the two, the peptide and the peptoid, bind our target molecule in the exact same position with a very similar affinity. But you said, wait a second, you also told us that they were doing a better job in terms of selectivity. I confirm that. In fact, we have used those peptoids variant of the IgG binding peptide. We have used those peptoid variants to separate IgG from the most complex source of IgG you can think of, which is animal serum. And look how good of a job they're doing from actually different sera, human, bovine, ovine, and again, our friends the camels. The source is very complex. But the product that we elute, the product that we recover from our porous media functionalized with peptoid ligand, is actually of excellent quality. And one thing that I want to show you is particularly interesting from ovine and camel antibodies, because you see here the goat IgG purified with protein A, which is the gold standard in the industry, doesn't actually come quite as pure as the one that we purify with our peptoid ligand. And even more interesting is the camel, because protein A, again, the gold standard in the industry for antibody separation, can't bind it. Our peptoid can bind it, and not only that, it binds it pretty well. It recovers it with a high level of purity. One last thing, I'll try to do it very, very quickly. one would say, well, sure, you talked about IgG. IgG is a very sturdy molecule. Everybody who works with IgG knows that antibodies are very sturdy molecules. They can be stored for a long time. They can be sort of mistreated. They don't lose their function. They don't lose their structure. But let's take a look at molecules, proteins that have been approved for therapeutic use recently by the FDA. You will see a lot of blood factors, blood coagulation factors, not as sturdy. They're actually very labile. One of them is even called the labile blood factor, just to give you a sense of how chemically weak that molecule is. So you cannot purify them through the classical affinity approach, bind and dilute, bind and recovery. Why not? Because to recover them, to disrupt the interaction with the solid phase, you actually need to mistreat somehow your protein. But if you mistreat blood coagulation factor, they will lose their bioactivity, and you will basically recover a product that's good for nothing. So our idea is, well, why do they have to be recovered using chemical agents? Why can't we recover molecules using light? Why can't we modulate the affinity between a ligand and a protein using light, using something that will not perturb the biochemical stability of the target protein? So that's what we started doing. So our idea is that we can engineer within peptides. You remember going back to the concept of Lego, you can put Lego of different colors into a construction. So once again, modularly, we can insert light-responsive moieties within peptides, within cyclic peptides, and make their structure change, and therefore make their ability to bind a biological target change using light. Something that a protein wouldn't mind, no matter how weak it is. So we had to find some sequences, we had to do some modeling to basically achieve this goal. Something that would allow to elute a protein by shining light or by cooling the solid phase. We came up with two sequences which were sort of borrowed from the literature. They're known to bind a protein standard, which is streptavidin. That's the only protein we have tried so far. These two sequences were known, but they were not light responsive. So we had to engineer the light responsiveness into that. And so again, lots of simulations to check what sequence would actually play the trick. And after we came up with some slightly modified sequences, we tested them. And again, instead of going through the numbers, I'll just give you the punchline. The affinity changes between 800 to 1300 times, going from no light to light, which means you can buy a protein in the dark, you can bind the protein in the dark, and you can recover the protein just by removing the cover, just by exposing it to infrared or visible light, blue light, which was, to us, particularly cool. So, to wrap up, and actually now Mike can be actually mad at me because I probably most likely stepped over my time, and that could be a reason for fighting. But are there conclusions? Truly, no. We just keep going, and we just keep building our Lego, and we just keep making it fancier and fancier. Why is that? Well, because first of all, because new problems come up: purification of labile proteins, engineering of nanotherapeutics. But mostly because peptides and peptide mimetics are a class that enable us to fantasize about solutions, because peptides are modular, because the building blocks that are needed to just get our creativity running wild are out there, commercially available. They just need to be put together. So the conclusion is we're just going through different analysis, and we go after newer problems with this tool, which basically has no end in terms of the things you can construct with it. For doing all these things, you need a fairly large group. So we actually, in collaboration with my previous former advisor and mentor, Ruben Carbonell, we actually put together a fairly large group that works on peptide. My group now has 9 grads that work on that, and a bunch of undergrads which are as good as the grads. And we have funding, both federal and industrial, that I would love to acknowledge now, because that's how we can buy the Lego. And NCBC for hosting us so graciously. Thanks.
your questions. We're going to have both speakers come back up after Michael's had his chance at the podium. So Michael Daniele did his undergraduate degree at Rutgers and then went to Clemson for his PhD, where he did polymer chemistry and nanomaterials. You want to go ahead and start?
Oh, actually, I actually did all the
Okay.
switching
He
already.
then went to the Naval Research Laboratory for his postdoctoral fellowship, and that's where he got into hydrodynamic focusing and tissue engineering. You'd never guess who he worked with there. And then I left, and he got promoted. So he was Isabella and Jerry Carl Fellow as a beginning staff member at the Naval Research Lab, and got into wearable sensors, making his own patches that would remind you that your heart really still is beating. He came down here. He was actually recruited by both the biomedical engineering program and the Assist Wearable Sensors program. The two department chairmen in B&E and electrical and computer engineering had a fist fight to see whose department would get him 51% of the time. And Dan Stansel's bigger than Nancy Albritton, so he won. But anyway, Michael will convince you that he thinks peptides are just another form of polymers. Stefano says those are fighting words, but we'll see at the end. Michael?
Thanks, Fran. So I don't know what it is, but apparently whenever we're involved, it instigates fights between other people and maybe between Steph and myself. So what I want to talk about you today is my BME side of the work that we're doing, but more, I'll call it from a philosophical bent. So this data-driven theme, how the work we're doing fits into data-driven drug discovery. And just some ideas I have, how our work fits into data-driven drug discovery, how it fits with Stefano's work on the peptides and potentially what he's doing with them, and how we could obviously make his work much better. And so we'll look at what I call human barriers. So these are hurdles on two fronts, from a technological side of how do we overcome human barriers, how I define them, and then really the big barrier in drug discovery, which is getting from preclinical to clinical testing to not only just something that's approved, but something that really works, right? Works in a large population or even just a defined population. And I think the human barriers are the biggest hurdles in drug discovery and drug delivery, and that's where some of the overlap work comes in. So what we'll roll through today is looking at, just kind of frame the picture, translating lab experiments or preclinical work into clinical trials, and where the stumbling blocks are there. Specifically the blood tissue barriers, which is what our group works on. Organ-on-chip models for these tissue barriers. So how do we circumvent some of the problems, overcome some of these hurdles by gathering more data, or how we can gather more data on these barriers. And then just the concept of data-driven translation into clinical trials, how we fit in, how the hardware that we build fits into drug discovery and drug delivery research. So if we look at the big picture, right? A lot of people are in the pharma industry looking at drug discovery, looking at drug development. We heard about the massive amounts of data that are involved. So we all kind of have seen the drug discovery pipeline or profile. You start with maybe some target. So going to Steph saying, hey, here's a biomarker or target we want to go for. Apparently we don't need Steph. We go to Google, 15 minutes. I could find this peptide interface. And then we could start there. Or we go to Stephanie and say, I've got this library. And now we need to screen a library. And he could tell us what's more selective, what's less selective. But I might have 100 hits, 1,000 hits. So that starts somewhere down on this side, right? You're looking at a marker, you're looking at a target, and you're trying to pair the two together. We do a lot of lab bench work, we do a lot of screening and cell culture, and we have this preclinical phase, right? So we have cells in a dish. I was a polymer chemist, so probably some part of me internally is heavily coated in polystyrene, but most people aren't, right? So cells in a dish is not a super applicable way of looking at pharmacokinetics. It may give you some hints, but I think it's informing some poor decisions, right? And we get this hurdle that comes up, or this barrier between preclinical and clinical trials, right? And I'll sort of lob a hand grenade right off the start and say that our starting hypothesis for a lot of drug discovery moving into clinical trials is wrong. So if our starting hypothesis is incorrect, any conclusions you draw from it are inherently incorrect, right? So we have cell culture informing animal models, then we have animal models informing clinical trials, and then we usually have smaller groups inform whether we proceed to larger groups of people or clinical trials. Well, cell culture doesn't really look like a full animal physiological system. And depending on, I guess, who you are, the animal model doesn't really look like the human model that it's trying to base itself off of. So one of our ideas, and kind of a broad swath of ideas now, is, okay, how do we overcome this hurdle? How do we fill in the gap? Maybe we don't necessarily get rid of animal models or get rid of some of these preclinical phases. How do we better recapitulate what the human model would look like? How do we generate the data or better data to fit into these models or to fit into this pipeline, right? Ideally, we would love to have human data along the whole pipeline. That's not plausible, right? You don't jump into clinical trials, so what can we do before that? And one of these major hurdles that we want to look at, so if we just narrow it down, right? We would like a full human kind of model that we could plug in and get data from and do variations. But maybe if we just took bits and pieces of a person or of the human model. And the bits and pieces that we look at specifically are blood tissue barriers. So if we think about drug delivery, it's probably going to be maybe some topical application we could think of through the skin. So we have a unique blood tissue barrier there. So you have the skin, so you kind of have this inherent tissue barrier, and then you have some connectivity to blood vessels right below that moving down. We have the blood-gut barrier, right? You're going to take some oral medication, tons of pills. So now we have some epithelial. So you might have an oral barrier if it's something that's going to be absorbed in the mouth, something in the intestine, something in the stomach. We want to get it into bloodstream. We want it to go synthetic. How do we know that type of stuff's working? And the one that we work on that I'll call the novel or the eccentric barrier is the blood-brain barrier, because that's kind of the pinnacle of physiological barriers. It basically keeps everything out, and when that's dysregulated, really, really bad things happen. So that's one where you could almost see, if we understand the fundamental barrier properties of that, how do we tune or how do we interface with the other barriers of the body? How do we get drugs into the brain? Not a really easy thing to do, not a practical thing to do. Right now, you know, you would go hopefully intravenous with some different liposomes they look at, but bad things get into the brain, right? You get encephalitis, bacterial, viral issues. They found a way across the blood-brain barrier. So what is it that's letting them do it? Can we mimic that, or can we use drugs, get them across there? All right, so these are three barriers that become challenges when you're screening drugs, or you're trying to select a drug, or you're trying to test a drug and go from preclinical to clinical. These barriers are different in animals. Barriers don't exist in cells on a plate. So how do we recapitulate them as human models? And so what we're working on are organ-on-a-chip models of tissue barriers specifically, right? So the organ-on-a-chip concept is if we could find the unit cell of some organ function and build it on a lab bench. So whether it's building the unit cell, kind of it has all the properties of a lung or a liver or the stomach or an intestine or a bone or the brain, in our case, the skin, gut, and blood-brain barrier. Can we recapitulate all those functions in a chip or in a device? And then say, this behaves just like that human organ. And this has been kind of an effort for maybe the last 10 years, right? Trying to build these devices the best we can to get from cells in a dish to what would be called a real organ on a chip. And they're great because they include human cells. So right off the bat, we're starting pretty good. We can include human cells and multi-cell types, right? So they're interacting, much like they would in the body and the barriers. Sort of high throughput, right? So not as high throughput as cells in a dish, but you could build a series of these, lower cost than animal models for sure, house them in an incubator. Right, here's a model of the liver that's in a multi-well format. That's good, we're starting somewhere, right? We got human cells, we've got high throughput, we can start doing testing. They have slightly more physiological relevance than just a monolayer of cells or even a co-culture of cells. But they often include what I call exotic materials. So here's a lung on a chip. It has an airway, an epithelial layer, endothelial layer below it that's would simulate the blood vessels, and there's a PDMS membrane across it. It's not very anatomically correct, right? So we have this PDMS membrane, and essentially if you were to try to compare that to the alveolar blood barrier that would exist natively, it doesn't work as well as a barrier. So although it's simplifying it, you might see a drug pass through, the airway into the blood, and you're like, ooh, look, it's systemic. But that barrier wasn't as good as what would be in an actual human model. So it fails to some degree. It lets a drug pass the screening stage, and then when you try it, it fails, right? So we're trying to remove those failure modes by better recapitulating the human model. So often we have incorrect anatomy. No one's liver is a monolayer. Think of the kidney, extremely tortuous, complex 3D structure, inherent in its function. You go back to any chapter 1 of a biology book, right? Form and function are usually very closely tied together. So we can do a lot of biochemical analysis. If we're looking at total function, it's sort of lacking. So can we build the microanatomy to more accurately mimic human organ function? Then kind of some stuff that's down the line. There's no interconnectivity, right? So your organs don't operate in isolation. So what you'd like to do is actually pair up these organ on chips in some fashion. So if you had some oral barrier that was connected to a gut barrier that was connected to a colon barrier, you might be able to track, maybe liver as well for metabolism. You might be able to track some really complex pharmacokinetics. And so I'll just toss it out there so you're thinking about it. That's a lot of data, right? That's going to be generated from those 4 chips in a sample just of N of 1 of what's going on across that path. And the bigger issue is there's no diversity right now, right? So you'll usually use, some people use cell culture lines. They're essentially homogenized cell culture lines. You may not have that same genetic diversity you're going to have in a clinical trial. So you're like, yeah, this looked great. This worked in our discovery process, but it turns out there was some effect down the line in the population diversity that it failed massively. This isn't just really isolated to organ-on-a-chip models. This pops up in a lot of animal models too. So in the lab, we get these genetically modified, genetically isolated mice or other animals, and you've essentially have this purebred defined model for X, and that's what you're testing your system against. So once again, your starting hypothesis doesn't account for this population. Yeah, you kind of found out one thing, but it's almost a false positive to some degree, right? If you have these large animal models that are almost segmented. So we can improve on that. So organ-on-chip models right now, they fail to recapitulate a lot of barrier functions. They're better than cells in a plate. They're not quite there for, I would say, replacing animal models yet. But that's where we want to progress to. So what we're doing in our lab, I won't dive into the technical details too much, but we're engineering these barriers. So the skin, the gut, the brain, we actually want to build these entire barriers on a chip and make them as biologically and physiologically relevant to the native system. So we figured we start with actually building the blood vessels. That's kind of uniform to all the devices, all the barriers. We need some type of endothelialized layer and blood vessel. And so what we're doing is we're actually building we took our engineering expertise and we decided to, okay, how do we produce fiber? And can we just produce fiber that's blood vessels? Same methods, right? A microfluidic shaping technique, it's an extrusion technique. We do some fancy chemistry that I won't get into. But essentially you could picture a metal device. At the end, we load in a bunch of cells, a bunch of materials, turn on a light, magic happens, like the peptide release, and you get this constant stream of microfibers out the end. And what they look like, so these are the walls of the fibers after about 10 days. Right as soon as they come out, they're filled with endothelial cells. We could pick, these are primary human cells. So we're already starting with the human stuff in our fabrication process. 10 days later, we have an endothelialized lumen, which you could see right here stained. Nice hollow lumen on the size scale of arterioles or venules. So those are 1 notch down from arteries and veins, slightly above the capillary. And this is a human microblood vessel. We can make meters of it. Incubate it, it's freestanding, you can move it around with your hands. It's fragile, like just about any tissue that's about 100 microns across. But what we have now is our primary barrier. There's no synthetic scaffold per se that it's sitting on. It has the appropriate microanatomy in terms of architecture, right? We have this hollow tube. It's not just a flat plane of endothelial cells. Endothelial cells that line all our blood vessels will behave differently if they're on a flat surface, flat hard surface, versus soft, squishy, round surface. So we're better recapitulating kind of that barrier function, which is this endothelialized wall. They have high chemoselectivity, right? Once you form these tight junctions, they form a nice barrier, right? You just don't have everything leaking out of your blood vessels, hopefully. And there's no passive transmission, right? That goes into drug discovery. So if we want to think about drug delivery agents, we now have a barrier function that recapitulates the blood vessel. We want to do intravenous injection of nanoparticles, liposomes, drug carriers, colloidal mixtures, right? Chemotherapeutic agents. How do they work their way out of the endothelial barrier? Can they deliver drugs? Do they still have their cargo? Does the blood vessel wall do something to the drug carrier, right? Are there some kinetics going on? Important questions we can now start asking in a more humanized system. So we got the blood vessels, right? We've got as many meters as we'd like. They're incubated. We could pull them out and we could start engineering tissue with them. So what we have is kind of this vessel on a chip model, or the manifold device, I might say. You take these blood vessels, you put them in a 3D culture of cells into a microfluidic device, and you start pumping fluids down it. Pretty simple. We could use different pumps, peristaltic pumps, syringe pumps, kind of fancy non-active pumps, pulling pumps, and different microfluidic devices. And what we can do is we could load basically all the cells in. We could have cells around our micro tube. So this is the microvessel here. There's just some red microparticles flowing through so you can see where the vessel is. Human dermal fibroblasts. They line up, they surround the microvessel. T0, after 10 days, I'm sorry, that's 4 days on this one. They're basically all lined up, lined along the endothelial layer. And we basically have that starting skin barrier. This is what the device looks like on the outside. Pop on human keratinocytes, dermal keratinocytes, that's the upper layer of skin. And that's what you see right here. So this model, if we took a cross-section, would have keratinocytes and a stratum corneum, which is basically your external skin barrier, the anuclear portion of the skin. Some people say dead skin, that top layer. Living keratinocytes, dermal fibroblasts, and then they contain endothelialized microvessels. So we have our human skin barrier on a chip. We take peptides, we take an array of peptides. Someone gives us lots and lots of money so we can build hundreds of these. And we start screening libraries in a humanized device that's 3D, that has a systemic component, right? This has actually two barriers. There's the external dead skin layer barrier, the stratum corneum. Then there's the endothelial skin barrier. So you can see if something is just absorbed into the skin or if it becomes systemic, which you may want in a topical delivery agent, or you may just want it localized in a certain portion of the skin to treat some type of dermal disorder. So we can start building these barriers. We don't have any exotic materials. Our scaffolds are biopolymer based. They're gelatin or collagen based materials. We could use other types of scaffold. We do extracted ECM and things like that. We have the appropriate 3D microanatomy. I'm working towards validating that. And we also have the proper fluid mechanics. So mechanics including flow as well as just fluid ratios. The amount of fluid that's present in the environment, we can tailor that and control that to match what would be physiologically relevant. And so we could do this. We could surround these blood vessels in astrocytes or glial cells. That's the blood-brain barrier. We can surround them in human intestinal crypts or stem cells, so it's a much more complicated process, but we could restructure that gut microanatomy that exists. We could do epithelial cells and try to recapitulate the oral mucosa, right? So if we wanted some type of delivery, that method. And so in terms of discovery and validation of drug carriers, what we look at in terms of our set is skin, gut, blood-brain barrier. And you have unique factors in each that you need to engineer into the systems, right? So in the skin, like I said, we have an anuclear barrier, so we have this top dead layer, right? So there's that barrier that we need to recapitulate and look at. You wouldn't get that in cells on a plate. It's much, much different in a mouse model or a dog model. Porcine skin's a bit closer. You're not gonna run through, right? You eventually move to porcine models. But it's not the same as the human skin barrier. We might look at topical delivery. Blood, gut, right? We have this unique epithelial, endothelial barrier. So once again, we have two barriers. So what's absorbed by the intestine, the epithelial layer, then what gets systemic, what becomes systemic. So we could look at systemic delivery, pop a pill, see how it moves through the system. And then the blood-brain barrier, like I said, is unique. So we could really play and learn with the blood-brain barrier how these barrier functions work, specifically in the brain, to develop novel drugs, novel compounds that will cross this barrier, which there isn't a whole lot in that research base right now. There's unique electrical connectivity that you don't see in the other barriers that you have to deal with or use to your advantage. So if you could recapitulate both glial cells and neurons, you can start controlling the blood-brain barrier potentially electrically. So we've all heard electroceuticals and play into this range. I call it a high barrier. It's kind of the extreme of the barrier functions in comparison to the other tissue barriers. It basically blocks almost everything out. All the models we currently have now, even the organ-on-chip models, are orders of magnitude reduced in terms of their function. So it's not very comparable. So you see something go through there, you almost take it with a grain of salt. Like, well, we affected the blood-brain barrier, we got something through, we have to do a lot more testing. A lot of things get knocked out along the way because as soon as you reach, so one measurement is called TEER, so basically transendothelial resistance. It's a resistance measurement like conductivity, orders of magnitude off in the models we create. A lot of times because there's some synthetic membrane in between, PDS membrane. This high resistivity is based on cell-cell interactions. And that's what we're going for. And so we could look at things like pathology. How do bacteria, how do viruses get past the blood-brain barrier? Yet we can't synthetically create stuff that does it very well. And so I'll leave us with kind of the data-driven translation. What happens and what you want to do is if you could build these models, and you could build 10, 20, build them on a well plate format, like a lot of tissue cell culture, you're generating a ton of data. So this isn't just cytotoxicity data. This isn't just genome data, right? You have 3D tissue samples. This is histological data. This is image analysis. This is process analysis. Does something change overall in the bulk tissue as you're operating these devices? You can also start varying the tissue. If we start with stem cells, then you're looking at development of tissue post just kind of the formation of the device. You could vary each well. You could do autologous stuff. So you could take samples from someone and not immediately jump into a clinical trial. And so we could simulate different variation, which is a big part of the trial process. I'm told Steph took all my time, so we'll move on.
Questions, and I have one, but I'll yield to the floor if we've got one from the floor. Yes.
Michael, in your models, you're putting the effort to make some blood vessels. Are they just sitting there, or are they actually being used to the fluid actually going through them?
So the fluid's actually going through them. So when we make the blood vessels, they're basically just the fibers containing the cells. We incubate them until they form a lumen. Then we put them in different microfluidic devices and actually pump fluid through. And so then we could polarize or better physiologically replicate what would go on a blood vessel. And we see a huge change in the performance of the cells inside as soon as we start shearing and creating fluid passage through them. And we think that's really critical for the barrier properties.
Us?
You mentioned that you created a meter.
Yeah.
You mentioned that lots and lots of money, and I can create hundreds of these. But on a commercialization pathway, that doesn't demonstrate manufacturability. So I'm curious to know from where you are, what you see the greatest manufacturability challenges, and if you have any insights on how you overcome those manufacturability challenges.
So I think in producing material, there's not a huge hurdle in terms of our processes. I think, especially with the blood vessels, it's scalable. It's a scalable process. The challenge, I think, is going to be in validation of the devices and essentially process control. You have a lot of living components, right, going into building these devices. So how at the end do you validate that device and say, this is what it is. This is a human XYZ performing at this metric. And that takes a lot of time and analysis.
Are you using validation in the FDA sense of using the validation, or
No.
in the sense that a scientist used the?
I think in the sense of a scientist, but in terms of quality control, if you picture this as a device, we are using validation in that sense of we want to validate it as this model, and then the quality control of, I put this number of cells in it, and we have this equivalent living cell population and genomic data and performance in the end product. And I think that's the bottleneck right now, is figuring out how you do that.
One of the things we've spent a lot of time talking about with many of our colleagues is how do you do sensing or imaging or some kind of monitoring of function in these complex multicellular three-dimensional systems without killing anything.
Yeah.
That's going to be a to get to where you're talking about. That's going to be a critical component.
It's essentially the need for inline analysis. I think is the biggest hurdle.
Okay.
I just have a question about scale in the other direction, which is, so you said you were able to make arterial sized vessels, which is amazing. I like this idea, but I was wondering, it seems like arterioles deal with a much different pressure environment than capillaries. And so are the properties, are you going to be able to easily scale down to, say, capillary size? And even if you couldn't, how different are the properties between the endothelium on capillaries and on arterioles?
The barrier functions are dramatically different based on just the physiology. We can scale up and down easily based on the fluidic fabrication. The other thing is what we expect to see, and I didn't show it, is we expect growth from these. So in these chips, you'll actually see angiogenesis.
Oh, wow.
And so what you'll get from that is potentially the capillary formation. So that's why we kind of settled in the middle. But the physiology between capillary, arterial, and manual is very different, especially in the barrier properties.
So I'm going to give Stefano a chance to answer a complicated question. I have a whole bunch of simple ones, but I'll ask you those later. If you were to continue this Lego theme, if you want to make a complex 3D system out of Legos that you could put in the body to recruit the right cell types to the right places, could you shortcut making an artificial organ or even an artificial tissue on chip by not having to physically put your cells in the right place, but having the chemistry of your peptides in this complex matrix do that for you? And if so, how?
Well, that's what that's what Michael and I are actually trying to do these days, or
Proposal?
proposing these days.
No.
We actually want to see whether we can direct cell migration and cell development on the surface of a chip. So we can basically utilize peptides to either display proteins or just the peptides on their own to direct the migration of cells through a pathway. So instead of flushing cells through a microfluidic, like an old-fashioned or let's say old-school microfluidic device, if there is old-school in microfluidics, but anyway, so through a channel, through a microchannel, actually let the cells migrate and differentiate phenotypically and build. The example that we have in mind is indeed angiogenesis, indeed formation of blood vessels, so that they migrate through their own inherent kinetics and not through a fluidodynamically controlled kinetics on the surface of a chip, so they can actually create a tissue in situ, a functional tissue in situ. Yeah, that's the idea.
I think we're out of time. I've got the signal, and we have